Hey everyone, this is Module 8 video, and I think I'm going to squeeze this into 2 videos, so I've sort of stripped this down to the basics just because we don't have much time. So what I want to cover in this video is, number 1, just some definitions and terminology having to do with mutation. And then a number of different ways to classify mutations. So I'll go over those and define those. And they're pretty straightforward. And you should be able to, give a definition for those or assign a mutation into a different type. One way to define them is spontaneous versus induced mutation. So the other part of this is to talk a little bit more detail about what causes both of those types of mutations. And I'll just run through a few examples of those. All right, so let's start with definition of mutation. A mutation is an inherited change in the DNA sequence of a chromosome in an organism. Genes that have just one allele are called monomorphic, and the allele is called the wild-type allele. If the wild-type allele is mutated, that's called a forward mutation, and the new mutant allele is called the new allele is called a mutant allele. You can also have the case where a mutant allele mutates back to the wild-type allele. That's a reversal, let's say, of the original mutation. So that's called a reversion mutation, or a reverse. Forward is mutation from wild-type to mutant. Reverse is mutation from mutant back to wild-type. So this is just showing if you start out with a wild-type sequence, GC, and it changes to AT, that's a mutant allele. If it changes back, that's a reversion mutation back to the wild-type allele. Okay, other ways to classify are by where the mutation happens. So it can happen in a somatic or body cell, and in that case it won't be passed on to future generations. Or it can happen in a germline, the gonad producing gametes, and that will be, or could be, passed on to future generations. Mutations can be classified by the type of DNA change involved. So one is substituting one base for another, and this is a base substitution here. G was substituted for A. Deletion, that would be removal of a base or a number of bases, or insertion is putting in addition, I should say, of base or bases into a wild-type sequence. Mutations can also be classified by the effect they have on encoded proteins, but I'm not going to cover that topic in this video or this module. So just to illustrate somatic versus germline mutations. Somatic mutation is in body tissue other than that germline, So it can affect the phenotype of the individual. In this case, fish, it could be a patch of color. In humans, somatic mutations are the main cause of cancer. So it's not that they don't have an effect on the individual in which they occur, but they're just not passed on to future generations. Whereas germline mutations are passed on, and the offspring would show the phenotype throughout its whole body. Okay, base substitution. So this is different types of mutation based on the change that occurs. So this is also called a point mutation. It's the smallest unit change you can make, changing one base to another. Okay. Now there are 2 ways you can describe this: a transition point mutation versus a transversion point mutation. A transition changes one base to another base of the same kind. So it changes a purine to a purine. For example, guanine to adenine, they're both purines, or vice versa. Or it changes pyrimidine for pyrimidine, thymine for cytidine. A transversion mutation is a base substitution in which one base is changed to a base of a different type. So purine to pyrimidine, or vice versa. Okay, so this is illustrating them. Transition defined here, transversion here. If this is the wild type sequence, and you have GC here, and that changes to AT, that is a transition mutation. When you make the comparison, you compare the top strand, G to A, both purines, C to T, both pyrimidines. If, however, you change GC to CG or TA, that would be a transversion because the G on the top strand is being changed to either a C or a T. G purine, C, T pyrimidine. Okay? And for insertion deletion mutations, if this is the wild type sequence, inserting a single base here, T, GTA becomes GTT, and the A is still there, but you have that additional base being added. And a base deletion is removal of a single base compared to the wild type sequence. Okay, another way to classify is by what are called spontaneous versus induced mutations. Spontaneous mutations occur without knowledge of what the cause was. It's not like there wasn't something that happened, but we're not aware of any intentional exposure to a mutagen. Okay? Spontaneous mutations occur at a very low frequency and are due to natural things that are going on. So these can be errors occurring during DNA replication, exposure to natural radiation that's present, always present in the natural background, or spontaneous chemical changes to the DNA bases. DNA is a very stable molecule, so that's a pretty rare event that would happen, any of those events. Now, there can be mutation in which it causes called induced mutation that occurs at a much higher rate than spontaneous mutation. Induced mutation is induced in response to exposure to some kind of substance, agent, called a mutagen. The definition is a little bit circular here, but a mutagen is defined as a substance and agent that increases the rate of mutation over the spontaneous rate. So anything that elevates that spontaneous rate of mutation is classified as a mutagen. There are examples of chemicals that can act as mutagens, and also radiation, high energy radiation. And we'll talk about each of those in the second section. This slide is just illustrating a famous experiment that was done in mice, in which they looked at mice with different fur colors and the gene that was known to control that fur color. And they raised a number. This was done in places that had very large mouse colonies. And they checked for the number of mutations and the number of gametes, or offspring essentially, that were involved in that test, that observation. And this was done for 5 different genes. And what they found was the spontaneous mutation rate, there was no mutagen exposure involved here, averaged about 11 mutations per 1 million gametes, or individual offspring. And that's per gene. So 11 mutations per million gametes for a given gene. In this case, gene involved in coat color of the mouse. So that's a pretty low level, pretty rare frequency, 11 per gamete, million gametes per gene. Well, what are the processes that are involved in producing these spontaneous mutations? So the main ones are listed here. Like I mentioned before, chemical instability of DNA, and these are mostly through instability of the DNA bases. I'll mention these briefly, what they are and how they can cause mutation. Background radiation, and this would be either what's called ionizing radiation, and that's a very high energy, short wavelength, X-rays, for example. And ultraviolet or UV radiation can also cause mutation. These cause different types of effects on the DNA, and we'll discuss what those are. DNA replication is a very, I guess I'm looking for the word, reliable process, accurate process. And so the error rate in replication is very low, but not zero. So there are a couple of things that can happen that are found to be sources of mutation. We'll talk about those a little bit too. Finally, there are known to be DNA elements that are able to hop around through the genome. These are called transposable genetic elements. And those can also be the cause of mutation if these elements move and insert themselves into a gene. I'm not going to discuss these in this video or this module. We're going to focus on the first three instead. So what are some examples of chemical instability of DNA? So one of them is called depurination. Another is deamination. And these are happening spontaneously, in this case, just because the bases are chemically unstable. This is what happens in depurination. It's depurination means loss of a purine, only the base though. Okay, so this is a DNA single strand here. We're only looking at one strand, and it has two pyrimidine bases, T and C, and a purine base, guanine. Occasionally, at very low frequency, this bond breaks, and the purine base is lost. The DNA strand is still intact, though, the backbone, but this base is missing. It's called an apurinic site. So when this happens, and this apurinic site here would be the double-stranded DNA molecule, and the purine was lost here. And let's say this molecule, the cell, is getting ready to go through cell division, so it will replicate this strand. But what does DNA polymerase put in? Well, if the G was there, it would put in a C, opposite. But there is no base there, so the DNA polymerase puts in a random base. And in this case, it's put in an A. The other strand would still be intact, and that could be replicated accurately, and that would be normal wild-type DNA, which would be A, CCG. In this case, however, now this is a point mutation that has resulted from that depurination event. If that goes on to in a second round of replication, then that mutation would be fixed in the cell. And this would be a base substitution point mutation. This type of base instability, depurination, can result in replacement by a transition or a transversion mutation. In this case, G, TGGC, was replaced by A, so that would be a transition mutation. They're both purines. But it could be G is being replaced by a T. So that would be a purine being replaced by a pyrimidine. That would be a transversion mutation. Okay, another natural process that can happen, spontaneous mutation involving base instability, is deamination of cytosine bases. And the chemical structure of the cytosine base looks like this. It has an amino group, and occasionally this bond breaks and this amino group is lost. And instead it becomes oxidized. It rearranges the bonds in the ring also. and you end up with this structure. This is uracil. So uracil is normally only found in RNA, but if you have a cytosine that undergoes deamination, you end up converting a cytosine base to a uracil base. So if that happened here in a base pair CG, okay, and so then you'd have a C being replaced by a U. And then the uracil, if it's replicated, this top strand, the uracil would, DNA polymerase would recognize the uracil and incorporate an adenine opposite. And then if this is replicated in another round, the adenine would serve as the template strand, the bottom strand here, and that would direct DNA polymerase to insert a T opposite. So you'd go from CG, deamination of C to U, replication once, replication twice, and you end up with TA. Deamination of cytosine always results in a CG to TA, which is a transition mutation, C to T. Okay, another thing that can happen with bases and instability is what's called base tautomerization, and that's shown here. Base tautomerization is a switch from one chemical type of a base to a different type. For example, for thymine, here shown, this is the normal, most common type of thymine. It's called the keto version or conformation of thymine, and the keto thymine base pairs with adenine. Two base pairs shown here. Very rarely, keto undergoes base tautomerization to enol thymine, shown here. And so this involves a single bond C to O and a double bond C to N. Basically, it's rearrangement of electrons in the ring. Okay? This enol version, OH, involves 3 triple bonds that would form with guanine. So if there's a switch from keto thymine to enol thymine during DNA replication, DNA polymerase would recognize this not as the usual thymine and not put an adenine opposite, it would put in guanine opposite. All right, so base tautomerization of T, shown here, tautomer version of T with a hash mark. DNA polymerase would put in guanine opposite. And then if the bottom strand is replicated in the next round, C would be incorporated. So you would go from TA to CG in 2 rounds of replication. Base tautomerization is always resulting in a transition mutation, just as shown here. T, a pyrimidine, converted to C, a pyrimidine. Okay, so those are types of spontaneous mutation due to instability in DNA sequence, especially in the bases. Okay, this slide is just showing that base tautomerization, although we talked about it for T in the last slide, this can happen for all 4 of the bases. It's a rare form for each of them, in which, if they're being replicated while they're in that form, it will result in a base substitution, transition substitution. Okay, one last topic related to spontaneous mutation, and that is mutation due to natural background radiation. And that can happen due to exposure to high energy rays, X or gamma rays, electromagnetic rays that are very short wavelength and high energy. Exposure to these causes breakage of the DNA strands. And so strand breakage, phosphate, backbone breakage, I guess, is another term for it. This can cause chromosome abnormalities, deletions of chromosome segments, losses of chromosome segments. So this is pretty severe type of damage. UV radiation can also cause damage to DNA, And the type of damage caused by UV radiation is not, it's not as higher energy as X or gamma rays, for one thing, but it does cause what's called thymine dimers, and that is when there are two thymines side by side on the same DNA strand, UV rays cause the two thymine bases to be covalently attached. And this produces a kink in the DNA strand, and if this undergoes replication before this can be removed, this will cause a base substitution as a result. All right, so one other mechanism is of spontaneous mutation is errors occurring during DNA replication. So these are extremely rare, and DNA polymerase does occasionally put in the wrong base opposite the template base. So if this is a G, DNA polymerase would normally incorporate a C. In this case, it's showing an A being incorporated. So that's a misincorporation. DNA polymerase has the ability to remove misincorporations almost all the time, but the rare case in which a misincorporation occurs and is not removed happens only less than one in a billion bases during a replication. So the human genome is made up of 3 billion bases. So that would be roughly 30 mutations out of 3 billion nucleotides that are misincorporated during every round of replication. Extremely rare. There is one other type of error that can happen during replication. This is called expansion of trinucleotide repeats. So some genes have trinucleotide repeats, meaning like 3 nucleotides CGG repeated nearby or within a gene. And these are 2 examples of diseases that are caused by replication-induced increase in number. So somebody with a wild-type version of this gene, Fragile X or Huntington gene, normally has 50 or so copies of this trinucleotide. But in replication, this can slip and the number can expand, and this produces the mutations which are going to impact, usually wipe out the function of this gene. These can happen in the coding region of the gene or in the regulatory region, resulting in basically null alleles. And this is just showing how a trinucleotide repeat can expand during replication. If this is a template strand and the DNA polymerase is replicating through this, making a product strand, occasionally the product strand can loop out like this, and DNA polymerase would just continue to extend from this 3' end. If that happens, then the product strand, if you start with 6 repeats and it loops out with only 2 repeats in the loop, you end up with a product strand now that has 8 repeats. So that can happen during replication, and expansion of trinucleotide repeats is a spontaneous mechanism of mutation. All right, I think I'm going to end this video here, and in the next video we'll talk about the other types of mutations and also what happens in induced mutation. See ya.